Thank you very much for introducing me and for having me here. And to start, I'd like to resonate what has been said before, but the trip has been made so nice by the good care of Peter Hedgie and also Agnes Bittera. It's been a pleasure to come here. It's been a very comfortable travel, and the coincidence was that I was on the same flight with Gábor Tamás yesterday, and he actually drove me from Budapest here to Szeged, and I learned a lot about Hungary, and also thank you for that. So when I thought about what I wanted to present today, I thought, it's of course humbling and very inspiring to be among so many established scientists that also inspired me throughout the years. But the energy I get in the lab is also from you, the youngsters. And you are going to shape the academic landscape in the next decades. So what I'm going to try to do is pass on a little bit of the passion that I got from Professor Sakmann to do science, and hoping that just the 20 minutes that we share in our lifelines, that I also pass on a little bit of that passion to you today. So this quote, "Doing science is fun," is actually a signature statement from Professor Sakmann in a volume of single channel recording, the book that he wrote together with Professor Neher. And he put this signature upon request of a Hungarian PhD student at the time in the lab where I was working. And Professor Sakmann would visit me every now and then to do experiments together. And the PhD student asked to have a signature, and this was the quote. "Doing science is fun." So when I think about this quote and what it means to me, it's actually manifold, right? So many of you may also have that scientific curiosity, right? And that is something that keeps me going on a daily basis. It's the scientific breakthroughs that are depicted here with cover figures in different journals. It's the scientific discoveries that fuel my academic spirit. In addition, I, of course, have been extremely fortunate to have such an inspiring mentor over the years. And through the postdoc I did in Heidelberg, I also have long-lasting international friendships, which I think are also very nice to be in academia, that you have so many international friends. And I've never thought that I would travel to Szeged, but this is also a very nice outcome of being in science. And the next layer I just want to briefly touch upon, science is something we do not do individually, but it's really through team science and with bundling the expertise of many people that we can really start making progress in academia. It's the rising stars, also in my group, that I have to acknowledge. The spirit that they bring to the lab is something I really, really enjoy. And what works for me is to have scientific heroes, just to reflect on the hard experiments and the struggles that these people overcome throughout the years, and I find that also a source of inspiration. So if you feel that that also works for you, right, then there are many examples here that may serve as a hero to you. So I'm going to touch upon two scientific projects briefly. And the first one is experiments that I did together with Professor Sakmann, and this was actually after I left the Heidelberg lab and had my own group. This does not mean that we stopped collaborating, but it meant that Professor Sakmann had to come by plane, fly into my lab, and we would do experiments together for a few days. And this is in awake behaving rodents. And what we wanted to try and understand is how different building blocks of the brain contribute to the representation of sensory information. And I can explain that here by that cartoon here. So every whisker on the face of the rodent is a discrete sensory organ, and it's connected to a module in the brain that we refer to as the cortical column, which is here depicted in these artificial colors. And the cortical column consists of different building blocks, the neurons or the different cell types, and we identify them based on the structure. Now, the rodent whisker system is a very nice model system to study because the whiskers are very discrete, right? You can identify every single sensory organ, but it also resembles a lot of how our tactile system works, right? So if you wake up in the middle of the night and you're looking for the bathroom, you don't switch on the light, but you use your fingers to navigate in your room. You find the doorpost, and the way we use our fingertips is very comparable to how rodents use their whiskers. The sensitivity of the systems are comparable. The anatomy in the brain is similar. We also use it actively, right? So if you want to feel a texture, you have to rub your fingers on the surface, and that's also how rodents use their whiskers, right? So this is what we wanted to understand using the rodent whisker system. So without going into many details, I did want to show you how we actually performed these experiments. So these animals are trained to tolerate being head fixed. We insert a recording electrode into their somatosensory cortex that is connected to a single whisker. We had a high-speed videography to monitor whisker motion, and the data then consists of an electrical recording of a single unit. So we refer to this as a loose patch recording. We obtain action potentials, also referred to in other talks as nerve impulses or spikes. We can extract them from the continuous trace, so we have individual spikes. We align this to the behavior. This is the whisker trajectory. And from the frame-to-frame analysis, we can actually determine when the whisker was in contact with the object, and we have then our touch windows. And eventually what we then want to do is align the activity to touches. So what we did here is we collapsed about 200 individual touch events and determined how the spikes align to the touch start. And as you can see in this particular example of this particular cell type, also represented here in red, that there is an increase in the activity. Now, to give you the upshot of this particular project, we wanted to focus on a particular event in the neuronal code, and these are high-frequency bursts. And Professor Neher already indicated when you go above a certain frequency, you get into supralinearities. So there tends to be more information in high-frequency events, and that means that the spikes appear at 100 hertz or more. So most of these events consist of 2, but we also find bursts of more spikes. And without going into the detail of all the brain processes that these events actually contribute to, it's just a very important part of the neuronal code. So to cut a very long story short, what we wanted to understand is how touches, whisker touches, are represented by different cell types. We took a coding strategy, meaning we looked at time in the experiment when there was a touch and whether there was spiking in different cell types. And we found out that the layer 5 thick tufted cells were the best coders of whether a touch happened or not. And you can see that here is the red part of the pie chart. That is the fraction of layer 5 thick tufted cells, this particular unit, that was a reliable coder of whether a touch occurred or not. We did it also the other way around. We looked at the burst events and determined whether a burst event was predictive of whether a touch occurred or not. And you can see from the blue line, those again are the data from the thick tufted cells. They are shifted to the right, and the further to the right, the best you can decode the behavior from the burst events. And also we used information theory to determine the information content of burst events, and we found out that there is more information in a burst than in similar number of spikes, but spread out in time, again for the thick tufted cells. So these were the experiments that we did together. In my lab, we also have a different research line, and I want to dedicate the remaining part of this talk to this. So we have been very much inspired and also influenced by the patch clamp technique. And in recent years, we started to collaborate with the hospital in Amsterdam to use this patch clamp technique on human brain tissue. And this means living human brain tissue. Now let me use this to carefully explain. So this starts with informed consent, right? So that the patients that undergo brain surgery actually agree that their brain tissue is being used for academic purposes. We get ethical approval from the relevant ethical board, and there's a special type of patients involved in this research. These are people with drug-resistant epileptic seizures, right? So they do not respond to any type of medication, and the only way they can be helped is when the epileptic focus is surgically removed from the brain. We focus then on patients in which the epileptic focus is deep in the brain, either amygdala or hippocampus, and the neurosurgeon then has to remove the non-affected overlying cortex to get to the deep pathology, and we get then the part of the healthy brain that is being removed in order to get to the deep brain pathology. And we treat that as if it's rodent tissue, right? The block that we get from the surgical theater actually has the dimensions of a mouse brain, right? So we take it to the lab, we cut slices, and we do patch clamping. So what we then can do is we can do single cell or multi-cell recordings depicted here. We dye-label cells to do post-hoc digital reconstructions. So after the tissue has been used for electrophysiology, we fix the tissue and we can do histology. And then all of a sudden this opens a spectrum of possibilities. So we had an excellent question this morning of how to relate intellectual capability to electrophysiological measures, but in a mouse it's very difficult to map cognitive ability, right? But in these human patients we can start pre-surgery with cognitive testing, right, or IQ tests. We can do gross morphology with MRIs, we can do action potential measurements, and we can do digital reconstructions. So the part I'm going to explain to you is work that we've been doing on human hippocampus, right? So in a subset of these patients, also the hippocampus has to be removed for instance, an amygdala-focused tumor. So here it's a slice where we show here in brownish that there are cell bodies. If you show this to a pathologist, he will say it's a normal-looking hippocampus. We do patch clamp recordings where we can run different kinds of profiles, and we can do reconstructions of the hippocampal neurons in these experiments. So the first thing that we noticed was that the human hippocampal neurons are huge, right? So perhaps you noticed in the rodent work that the thick-tufted cell was the biggest one. But when you show it side by side on the same calibration, then the human CA1 pyramid is much bigger compared to the biggest red layer 5 thick-tufted one. And also when you compare it to the same cell type but from the red, it's much bigger. Now this has implications for how these neurons work, right? And I just picked here a KLM flight because as a Dutch person I typically fly KLM. But when you have a KLM huge plane compared to a small plane, you know, it's not difficult to imagine that these things work differently. And it's the same with brain cells. The bigger they are, the more computational complex they are. So I show here a gallery of different morphologies. Of course, there's biological variability. We see that on average, all the dendritic segments where information arrives adds up to about 18 millimeters. And these are the levels that we find for the rat CA1 pyramids or mouse CA1 pyramids, right? So it's almost doubling in the terms of dendritic morphology. Now, I show here 3 different compartments in different colors. So we noticed that you have basal dendrites, we have oblique dendrites here, and then the apical dendrite here. And the reason why we distinguish the different compartments is because it's very well known that the major input to these neurons arrives on the oblique and the basal dendrites in hippocampus. So this is a drawing from hippocampus by the Hungarian anatomist Károly Schaffer, and those are named the Schaffer collaterals, right? And those axon projections are implicated in memory formation. So we, as a first step, wanted to understand or map the structural properties of these hippocampal neurons in human. So in the pie chart, you can see that the fraction of these different compartments add the same to the total dendritic length of these neurons. But what I try to capture in this scheme is that the organizational principles of the oblique and the basal dendrites are completely different. So for the basal trees, we see a few number of original trees, but for the many nodes, so branch points within a tree. And for the obliques, it's the other way around. Many primary trees, hardly any branching. And this is then quantified in these box plots, where you can see almost an orthogonal principle in terms of organization. So in this work, we started to collaborate with Edon Segev, and I'd like to refer to another quote that Professor Sakmann at that point also shared with Edon Segev, right? So I know a lot, but I do not understand, right? So let's use a model. And in this case, it means, you know, when we map all these organizational principles and we have knowledge about the structure and the differences between obliques and basal structures, but what does it mean for the function of the cell? And that is what Edon Segev then contributed. And he is a famous modeler from Israel, and the short pitch to the modeling effort would be, is the more obliques you have, depicted here as all these individual non-linearities contributing to the computational power of this cell at the end, the more obliques you have, the more powerful your CPU. And this also goes back to what Professor Neher already alluded to this morning. If you have heterogeneity, it expands the capacity of information processing. And it works the same for these human hippocampal cells. The more obliques you have, the richer the computation that these individual cells can perform. All right, so when working with Professor Sakmann, I learned over the years that you cannot stop when you have your plot, right? So when I would discuss my data with Professor Sakmann, he would tell me, you know, it's all very nice, but what did we learn, right? So that also resonated in my ear when I prepared this presentation. So the take-home message is burst events matter, right? So when we look in the rodent sensory system, burst events carry a lot of information. Second, from the second part, the human CA1 pyramids from hippocampus, they are really supercomputers. And another thing that I learned is, you know, you have to celebrate your successes, right? So doing science, and especially when you're doing difficult experiments, then those eureka moments are really, really nice to celebrate. And this is upon acceptance of the work that we did on the rodent burst event. And Jean-Pierre did a lot of the modeling work or the information theory calculations, and we celebrated this with a bottle of champagne when the paper was accepted. A few more slides. So I have really a special thanks to both Professor Sakmann and Christiaan de Kock for their mentorship over the years and for inviting me here, and I really cannot thank you enough for everything you've meant to me over the years. And then finally, I also have to acknowledge the people that are doing the experiments in the lab. So what I did not mention is when we get the tissue from the surgical theater, we keep the tissue alive, but there's a very narrow time window, right? So we can keep the human tissue alive for about 18 hours. So that means when the neurosurgeon resects the tissue at 10 in the morning, we have until about 5 or 6 in the morning, you know, and the people really work through the night. So they really deserve a lot of credit for their experiments. I also have the support and the funding agencies here. The Heidelberg family, meaning all the postdocs and the technicians and so many people that I would have 3 slides actually to thank everybody. But above all, please remember, doing science is fun.
So thank you very much for your very, very nice and informative talk with lots of personal details. I think that was really important for the students to understand better how important these personal connections are in addition to scientific knowledge and research. Now we would like to give you the certificate.